Hello everyone, welcome to my YouTube channel. My name is Farah Maarfi and I'm excited to be here with you today. In this video, we are going to be discussing the basic principle of sedimentation, a pillar on which centrifugation works. Before we get started, I just wanted to mention a few quick things. If you are new here, be sure to click the subscription button and turn on notification bell so you won't miss any of my future video. Don't forget to like and share this video with your friends and family if you found it helpful. Let's get into it. First of all, we should understand what is centrifugation. So, it is a process. It is a process that uses the centrifugal force to separate and purify mixture of biological particles present in a liquid medium. That liquid medium may be water. It is a key technique in isolating and analyzing cells, subcellular fractions, supramolecular complexes, and macromolecules such as proteins and nucleic acid. So, if we talk about centrifuge, then it is an instrument by which we can separate out these particles from suspension. Suspension may be any buffer, maybe water also. The process of centrifugation relies on the principle of sedimentation. Centrifugation relies on the principle of sedimentation. Sedimentation is the settlement of any particle under the influence of gravitational force. So, the principle of sedimentation based on the influence of gravity on object with different density. If the object have different density, the influence of the gravitational force will be different on each object depending upon their density. In essence, sedimentation is all about gravity acting on particle suspended in a fluid. Fluid may be water, causing them to settle based on the density and size. Let's understand this with an example. Suppose we have container. This container we have filled with liquid. That liquid may be water. And when we suspend some denser object, these black dots are the denser object. Denser object means these are having density greater than the density of water. So we suspend these denser particles into a water containing in any container. Then these objects will experience a downward pull, due to gravity. And this gravitational force make the particle settle out of the suspension like this. These heavier particle will settle down first, and then the larger and the smaller particle will settle out differently, and the settling rate depends on the size and shape of the particle and the density and viscosity of the fluid. So for the particle, the governing factor is size and shape. Size and shape. And for the liquid in which it is suspended is viscosity and density. Viscosity and density. So these are the four factors that decide the rate of settlement of the particle present in any liquid. As we see here, the larger and the denser object experience a greater gravitational pull and sink faster compared to the smaller or less denser molecules, or you can say the particles. When suspended in fluid, example water, while in this figure the container containing more viscous fluid, that is honey, when kept at rest condition, then under the influence of the gravitational pull, these particles or the object will sink slowly than those kept in water, that is having a less viscosity. Because the viscosity created a frictional force against the gravitational force, so that hinders the settlement of the particle. Since this is very slow process and is only applicable for the particles having a greater marginal density than the medium in which it is suspended. But if we want to apply the same principle for the biological sample, then we need to add some external forces. That force may be the centrifugal force. So why we need centrifugal force for the biological samples? Actually, the centrifugal force increases the rate of sedimentation and especially essential for the particles which have very less density or the particles that have density very much close to the density of medium in which it is suspended. So our biological samples have very less density and will exhibit drastic increase in sedimentation rate when they undergo acceleration in the centrifugal field. Let's understand this by an example. Suppose we have two buckets filled with water and crude sand particles were added to it. The particles will travel slowly to the bottom of the bucket by gravitation, but sediments much faster when the bucket is swung around in a circle. And when the same phenomenon is applied to the biological particle and was swung around in a circle, then the particle follows the path of circular motion and experiences a velocity that is constantly changing, and at the same time it also experiences the acceleration. So when these two things exist, then there is a force occurs that holds the particle in a circular motion and is always acting towards the center, and this force is called the centripetal force. And this force is always holds the particle in a circular motion, and opposite to this force there is another pseudo force that exists or the particle is experiencing at the same time, and this force is called the centrifugal force. The centrifugal force is responsible for the settlement of the biological particles when rotation is applied. This image showing how the centrifuge looks alike, and this is the cross sectional 2D view of the rotors that holds the sample. These are the two holders, and when the tube containing the sample put it in a holder, it looks like this. This is the tube, the gray color showing the tube, and it contains the biological molecules and some medium into it, and the particle here follows the same circular motion like this here discussed before. And the particles experience a rotation when some speed is applied to the rotor, and due to the speedy rotation the pseudo centrifugal force is felt by the particle inside the tube. The centrifugal force here drags the particle away from the axis of the rotation towards the bottom of the tube, and the particles will settle down here like this under the influence of centrifugal force. So this was whole about the principle of the centrifugation and the sedimentation. In the next upcoming video, we will see the centrifugal force and the principle behind it in a more advanced form. We will also derive the some mathematical expression of it, and after that we will have the theory of operation, and further we will see the types of the rotors and the types of centrifuge. So if you like this video, please like and subscribe. Thank you so much.